I'm personally a big fan of pasta, so much so that I decided to do my PhD on how to make spaghetti. I study viruses, but at its core, a virus is just a box of spaghetti. That's all it is. It consists of this outer layer that's called a capsid, which surrounds and protects the important stuff on the inside, the viral genome, which is what contains the instructions for making new viruses. Now, I study a particular related group of viruses that are called flaviviruses. And some flaviviruses that you may know are dengue virus, Zika virus, West Nile virus. Together, these cause about 400 million infections and about 10,000 human deaths every year. When flaviviruses get inside your cells and they start to replicate to make more of themselves, they all do it in the exact same way. They set up two locations inside your cells, one of which is responsible for making new viral genomes, new spaghetti. We'll call that the spaghetti factory. The second location, which we'll call the packaging facility, is responsible for making empty viral capsids, empty spaghetti boxes. And what happens is newly made viral genomes are sent from the place where they're made to the place where they're packaged. They're put into those empty viral capsids, and then now complete viruses are sent out of the cell to go infect more cells. Now, because all flaviviruses work this way, if we can find a way to interfere with this process, we may be able to simultaneously stop the replication of a wide variety of these medically relevant pathogens. My research focuses on the link between the place where viral genomes are made, the spaghetti factory, and the place where they're packaged, the packaging facility. We know that these two things are actually pretty close together inside cells, and it sort of makes sense, right? If you're making spaghetti in New York, you don't want to package it in Los Angeles, right? But up until recently, we weren't sure how the virus maintained this close connection. My research has discovered that there is a protein which is responsible for linking these two locations together like a chain. Now, I'll back up a little bit. A protein is just a tool that your cells use to accomplish tasks, and your cells actually have a linking protein inside them. It seems that when flaviviruses infect your cells, they steal this protein for their own nefarious purposes, namely linking these two locations. Now, there is some good news. My research has shown that you can actually delete this protein, get rid of the chain, and the cells are actually okay, but the virus is not. Flavivirus replication, when you delete this protein, is reduced by about 90%. I've also shown that you can mutate this protein or change it in a way that inhibits its linking ability, so breaks that chain. When you do this, again, viral replication is reduced. The protein is no longer able to help the virus replicate. Finally, I've shown that you can actually target this protein with a drug that inhibits its linking ability, and when you do that, again, viral replication is reduced. These results suggest that the linking function of this protein is critical to the replication of flaviviruses, and that interfering with this linking function may be a great way to leave a wide variety of flaviviruses cooked. Thank you.